Hello, this is Amanda Welch welcoming you to this Bite-Size Bio web seminar, which today is sponsored by QIAGEN. QIAGEN's a Netherlands-based holding company is the leading global provider of sample-to-insight solutions to transform biological materials into valuable molecular insights. QIAGEN's sample technologies isolate and process DNA, RNA, and proteins from blood, tissue, and other materials. Assay technologies make these biomolecules visible and ready for analysis. Bioinformatics software and knowledge bases interpret data to report relevant, actionable insights. Automation solutions tie these together in seamless and cost-effective molecular testing workflows. QIAGEN provides these workflows to more than 500,000 customers around the world in molecular diagnostics, human healthcare, applied testing, forensics, veterinary testing and food safety, pharma, pharmaceutical and biotechnology companies, and academia, life sciences research. Today's presentation is titled Common Challenges and Solutions to Microbiome Analysis Workflows and is being presented by Dr. Sven Reister from QIAGEN. Sven studied biology and biochemistry at the University of Dusseldorf, focusing on stem cell biology and epigenetics. He did his PhD thesis in the Department of Pediatric Oncology, Hematology, and Clinical Immunology, analyzing the gene regulation and expression of stem cell markers in hematopoietic stem and leukemic cells. Sven is currently working in the PCR product development team, focusing on the development of PCR master mixes to improve microbiome applications like 16S sequencing. As always, we will have a question and answer session after the presentation. So please type any questions that you have into the questions box, which appears on the right-hand side of your screen, and I'll put them to Dr. Reister at the end. The recording of the webinar will be available at bit.ly/microbiome analysis webinar. That's microbiome analysis webinar, all one word, lowercase. So now over to you, Sven, for the presentation.
Thank you very much, Amanda. So thank you, everybody, for joining, and have a good day. As Amanda said, I'm working at QIAGEN, and I'm working in particular on solutions and PCR solutions for microbiome analysis. And this is a more and more important market for QIAGEN. Microbiomes are getting more and more into focus because the interaction between the human beings and microbiome, as well as all other mammalians, plants, and the entire Earth microbiome is getting more and more into the focus. So I would like to start with a bit of background first. So what about the microbiome? Where do we come from? What is currently the situation? And also, what are the challenges you have to get from a sample to an unbiased result, because it gives you no benefit if your results you get out of NGS, for example, are not displaying what you had in your sample. So let's start first with us, the human superorganism. Looking at the cataloging efforts which were done in the NIH Human Microbiome Project, approximately 10,000 different organisms live with us, meaning on our skin, in our gastrointestinal, in our mouth, and on the entire body. The number of bacterial genes outnumbers our by the factor 150. And the different microbiota which you have on your body can now be revealed due to better technologies, and they show that they have an effect not only on us, but also our age, our gender, our weight, or our total habits, like dietary habits, have an effect on the microbiome, on the other thing. So it's a balance, and it's a back and forth between us and our microbiome. So why is it coming up now, and why is it something which is not obvious since years? Before molecular biology technologies were applicable for microbial analysis, you could only use things like culture assays to find bacteria. On the one side, this allows you only to detect and identify bacteria, which you can grow. And on the other side, you will just find what you're looking for. So for example, selective media for some bacterial strains are not suitable for growing other strains. So this was a very narrow view. And now, due to all our molecular biological techniques like next generation sequencing and also whole genome sequencing, we can have a better look in all this. And all these molecular technologies already have revealed some major impacts, so for example, the dysbiosis or imbalance in the microbiome can contribute to various diseases and also very severe diseases of the human being. When we look here on different areas of our body, in the gut, for example, the microbiome is associated with intestinal infections, obesity, inflammatory bowel disease, type 2 diabetes, or cancer. Also in our airway, bacteria are known to be responsible or have an impact in pneumonia and other respiratory infections, in COPD or in cystic fibrosis. Also, the urogenital tract is quite massively affected by bacteria, so you can get bacterial vaginosis, urinary tract infections, or sexually transmitted diseases. Obviously, as each and every other organ, also the blood can be affected, leading to sepsis or bloodstream infections. The oral cavity is also known and often found to have imbalanced microbial communities. In these cases, they often lead to periodontitis or gingivitis, so also perhaps not the most severe malignancies, but still something you have to suffer from. So as said, in earlier days, we just had the culture techniques or have done some tests for catalytic activity of bacteria to identify their metabolic activity and the characteristics to narrow down which species you have there. Nowadays, we have other options to identify the community in a microbiome sample. You can look either on the nucleic acids or on the genome. You can look out at RNA to analyze the transcriptome, or you look on proteins and other small molecules to look for the proteome or metabolomics. Every of these methods has advantages and downsides. So looking for the molecules is, of course, an interesting point to see what is predominantly secreted in regards of small molecules or proteins. Looking for the RNA expression shows you which genes are upregulated or strongly expressed in your microbial sample. So gene expression is an interesting point, for example, also for challenging or for tackling of infections, to find new targets for an antibiotic therapy, for example. Currently, the standard workflows to identify the diversity of a microbiome sample is to sequence the DNA. You can do this either by DNA sequencing of a shotgun library. This will give you a lot of answers, and this is not restricted, for example, to bacteria. You can have a look for bacteria, archaea, viruses, and all other DNAs in one run. That's on the other side also the point which is perhaps not optimal. So if you have a lot of human DNA in because you have a stool sample or something, you will also sequence your human sample. This leads in the end to the need of having a high sequencing capacity per sample. So you have to do really high coverage sequencing. You have a lot of data because you not just have a short amplicon, you sequence entire genes, entire genomes. It gives you a lot of additional information, but sometimes you just want to know how is the distribution of different strains. This information is often analyzed by using the 16S rRNA gene from bacteria. This gene is expressed or is inherited in all bacterial strains in different copy numbers. So you can have between 1 and more than 10 16S copies per genome, depending on your bacterial strains. The beauty about 16S sequencing is that you have a short amplicon, which is normally something like 300 base pairs when you look at the V3, V4 region, but still has a high discriminative power. So you can sequence this, have a low sequence cost in the end, and can sequence a lot of samples and get your bacterial diversity, so your OTUs and the composition in your sample. 16S sequencing will give you answers about the bacteria and the archaea in your sample. You could combine 16S sequencing, for example, with ITS, and by doing ITS sequencing, you would also get fungal and yeast strains in your sample. But during my talk, I will focus on 16S sequencing in contrast to the DNA shotgun sequencing approach, because this is the most common approach at the moment. For example, when we look at the Earth Microbiome project, with this specific protocol, more than 2,000 publications were made in the last years. So when we have this from the microbiome sample to your results, what are the challenges you have to tackle and you have to think about before starting it? Obviously, you want to sequence DNA, so you first have to find the perfect or suitable solution to extract high purity, high quality, and high molecular weight DNA from your sample. The extraction of the DNA should be quantitative. This means when you have different bacteria in your sample, you should be able to crack and lyse them all with same efficiency so that you're not getting a bias due to lysis. As said, fungal sequences are getting more and more into focus. 16S was the start, but now the research is more and more interested also to see what happens with the fungal microbiome. This means your lysis protocol or your kit is not only needed to be capable of extracting bacterial DNA, but also fungal DNA. And the better the technologies, the higher the demands of the researchers are. So 16S sequencing really became a high throughput application with a lot of samples every day in labs around the world. And also the requirements for the sample input are getting more and more sophisticated. So the researchers want to check not only samples with high DNA input like stool samples, but also want to look, for example, for skin swaps or something, or buccal swaps, where you not always have the most DNA, but still need to get enough out for your sequencing workflow. So after having your DNA extracted, you want to amplify it. So you have to identify a good PCR solution, which quantitatively amplifies the different sequences. Bacteria can have between 25 and 85% GC content in the genome. So finding an appropriate enzyme which amplifies low and high GC with the same efficiency is a key point here. In addition, especially when you have low input samples, you should have a chemistry which is not giving you 16S DNA background, because this again can bias your results. Obviously, if you don't have a lot of sample, your mix has to be powerful and sensitive enough to amplify 16S also from minute sample amounts. As said here, fungals are getting more and more interesting. Therefore, the demand is raising that you can multiplex, for example, 16S with ITS so that you can amplify your bacterial and fungal sequence of interest in one reaction. So now you have done all your wet lab work, you have given your libraries to the sequencing facility, and then they come back to you with something like 20 or 60 or sometimes even more gigabyte of data, which for me as a biologist or biochemist is something at the beginning I had no idea what to do with this. It's really a different world. I don't have a bioinformatics background, but you need this, or you need to have a good solution. Bioinformatics are important because you have to analyze more and more data. You have a higher sequencing coverage. You multiplex your samples so that you have 96 or sometimes even more samples per run with different barcodes on it. And in addition, you want to get all artifacts removed which are coming from your workflow. So ideally, PCR duplicates or something in this regard. You need a good trimming of your results. And later on, you just don't want to have pure data tables. You want to have them visualized in a good way, ideally usable for a publication or for the next meeting with your lab to present what you're doing. The last point is you don't want to wait 2 weeks for your NGS analysis, so there is the need to have a workflow which has a short time to result and to give the results you need. So that's the challenges before we start into the DNA extraction and amplification. I first would like to have some words on good working practices. When we started, or when my team started with working on low DNA master mixes for microbiome applications, we first had to find ways to set up PCR mixes without contaminating them on our own. So we are, in the end, the biggest contamination source. Each and every one of us is releasing around 1 billion bacteria per hour in the air around you. So we have a microbial cloud just around us. So we have to find ways to get this microbial cloud not entering our sample tubes. Some ways we are using are obviously dedicated disposables and pipettes to use suitable reagents. So also at this point to exclude that the reagents themselves are adding any additional DNA background. Running non-template controls is kind of mandatory because it shows you directly if your reagents are clean or not. And if you can do so, increase your sample amount and reduce your PCR cycles, because the background will be lower if you're not amplifying it that much. The last point I can tell you is that we are using a UV PCR bench for setting up all this, and by this can completely exclude contaminations, which I can show you later on too. In addition, we have identified, or we've checked that depending on your sample prep kits, you can have more or less DNA background. Therefore, this is also a good point to look for when you're picking your workflows. And as a last point, store your samples before nucleic acid extraction as well as your extracted DNAs appropriately. So let's jump into the DNA extraction part. So what is important for your samples? We said already that you require good nucleic acid quality. You don't want to have shearing. You want to have a reproducible isolation of high quality nucleic acids. For this, you have to be sure that you always have a sufficient cell lysis. You want to exclude co-purification of PCR inhibitors because when you have them in a sample, they can affect your entire workflow later on. And yeah, all bacteria or all germs in your sample should ideally be lysed and isolated using the right kit. So let's start with the first point. What is the effect or what is the source of PCR inhibitors? A lot of microbiome studies are working with soil samples or stool samples or other really complex matrices containing bacterial DNA. In soil samples, for example, you can have humic acids. Humic acids are known to bind directly on the PCR enzymes and by this inhibit the Taq polymerase. In stool, for example, you can have hematin. Hematin is coming from blood and is perfectly complexing 2 times positive ions like iron, which is its job, but it's also doing exactly the same for magnesium. So this is also affecting the PCR by complexing your magnesium. How does it look when you're doing an inhibitor removal or you're not doing it? So we've used challenging sample materials. In this case, it was a stool sample. You can see when we use the inhibitor removal technology, which is patented for QIAGEN, you have a really good amplification and you don't see any inhibition. When you leave this step out, you can see that the same samples have significant CQ shifts. Here, for example, we have something like 20 CQ shifts, and yeah, this is less than 10,000 times less amplified DNA. So the kit or the PowerCell Pro kit, which is containing, for example, this inhibitor removal technology, is not only giving you the security to have no PCR inhibitors in your sample. We also compared it with different stool samples and other vendors, and we can clearly indicate that we have the highest yields out of your sample. This means, on the other side, if you see it that way, to get more DNA out, you have to lyse more cells. And this means this kit is capable of doing or giving you a much better picture of your microbiome, which is in your sample, than other kits are capable to do. Here on the right side, you can also see this by looking at an alpha diversity plot. The PowerCell Pro kit has the highest diversity of all analyzed kits in this setting. So this is the first step you can go to certify that your microbiome sequencing approaches give you the best picture of your real microbiome sample. Think about what's your sample, think about the appropriate sample extraction kit, and yeah, this is already giving you a good basis to start with. So let's go on. As we now have our DNA extracted, we want to jump into the PCR part of the story. So for 16S, the challenges are that you have to amplify all bacterial 16S sequences evenly. This means, for example, when you use a mock community which you can buy at ATCC with 10 different bacterial strains, and each of them has 10% abundance in your sample, your NGS results should also come up with something like 10% each. Beside this, and this is also a point which affects your sample distribution of the different strains, your chemistry itself should not introduce any bacterial background to your sample. This is not always that easy because, for example, a lot of enzymes which are used for molecular biology applications are expressed in bacteria. And further on, you have to have a chemistry which is sensitive enough to also amplify limited sample amounts. You might think when you do a surface DNA extraction from soil or something that you always have a ton of DNA in. The problem is you not always know if this is pure microbial DNA. It can also be a lot of plant DNA, a lot of mammalian DNA. So when you end up with putting 1 nanogram of DNA in your sample, it does not necessarily mean that you have 1 nanogram of bacterial DNA. So you have to have a sensitivity from the mix. And in addition, as I said some slides before, multiplexing of different targets would be interesting. Here I'll show you data which we have generated with different solutions which are quite abundantly used in microbiome applications. You can see that the mixes on the one side are getting very, very weak for low input amounts. So with 2 E. coli genomes per sample, you're not getting really strong bands anymore. And the non-template controls can be even stronger than your low concentrated samples. So this means such a mix could introduce a DNA bias in your sample, which you can't evaluate. Here in another solution, you can also see that the antices are positive and could give you some input. I have to say these data were made with 36 cycles of PCR cycling, so the experiments are already intended to get down to low DNA amounts. We have developed a solution to tackle this. Our new QIAGEN UCP multiplex master mix is tested for bacterial and fungal background. And it's not only the master mix which is tested, all components of the kit are tested. Because we have found that using an appropriate water, for example, is also a key point. So not only the mix, the tracers we have in the kit you can use to color your sample and you could use to color your master mix, as well as the water, are tested with this test for bacterial background and, of course, fungal background. Beside the low background, which is already a key point of this master mix, it is a high concentrated formulation. So it's a 4x master mix, which allows you to add high sample amounts. We have customers doing a 20 microliter reaction, and out of 20 microliter, 40 microliters are sample, 1 microliter is primer, and 5 microliter are the master mix. So this is allowing you to, or enabling you to add big amounts of your samples in case they're not high concentrated. In addition, I'm not showing this here, but I have sequencing data where we have sequenced mock communities, and this mix is highly quantitative. So we came out with the bacterial abundances which were added to the kit. So that's the one part of the story. The other point is we have a high inhibitor resistance. So in case you're using extraction kits without inhibitor removal technology, like our UCP QIAamp family, which on the other side has a host DNA depletion, also a good point depending on your sample material, and we have a high GC coverage. So we could show that we can amplify from 25 to 85% GC without any modifications of the protocol or the mix. So how does points like an inhibitor resistance or the background come into play for your samples? Here we have used humic acids, a known inhibitor which is co-purified from soil samples, and looked for the 16S amplification. Here you see 16S with 220 or 2 E. coli genomes in the presence of humic acid and in the absence of humic acid. I agree, you have a weaker bands in the presence of humic acid, but you still get down to 2 E. coli genomes. Other solutions here are not able to amplify anything in the presence of inhibitory substances like humic acid. And even without humic acid, I have to say I can't discriminate non-template controls, 20 genomes or even 200 genomes, saying that the background in this master mix is quite high. And I got feedback or confirmation for this from customers we talked to, which see for PCR solutions, depending on the mixes, up to 200 bacterial genomes per reaction background. So that's the basis, and I would like to show you something what you can do when you have more samples or when you just don't want to spend your time standing on the bench and doing your workflows. So a lot of our kits, like the Kaya Amp 96 Power Fecal QIAcube kit, or also the PowerSoil Pro kit, which is coming now as a QIAcube HT version, are automatable on a QIAcube HT. In addition, all our spin column-based kits can be automated on a QIAcube in a 12 reaction format. So here, these parts of the QIAGEN automation do a lot of the part for the sample preparation. Here you have the lysing station to do the beating of your samples. You transfer your lysates after inhibitor removal to the QIAcube HT. It does the full sample prep of 96 samples in 90 minutes. Then you can use your extraction, your output plate, export your results file, use it here for the QIAgility. That's our small and really powerful PCR setup robot. You put the export sheet from the QIAcube HT to the QIAgility and directly have your sample list. So it will know directly in which position is which sample, set up the PCR, and in the end you get a report. And with this report, you know my sample, my stool sample from this patient is at position A1. After the PCR, you can use your plate and put it directly in the QIAxcel, which is a capillary electrophoresis system. But the good part is the system works directly from your primary sample plate without doing anything to your samples. So this means you can put your plate in, just look if you get your amplicons, and directly proceed downstream to your amplicon purification and, for example, the one-step library prep. Why I'm telling this to you? Because if you have 96 samples up to this point, it might not be that cost intensive. But when you go into the NGS workflow, you want to be sure that all your samples have amplified and have appropriate amounts of PCR product. So the QIAxcel can be perfectly implemented in your workflows at this point. Okay, so showing these pictures is one point. I would like to show you some data. We have used frozen stool samples, 200 milligram each from 16 patients. The samples were extracted with the workflow I've shown you on the slide before, with the tissue lyser and automated on a QIAcube HT. So the technician had something like 60 minutes free time, which he normally would have been on the bench and putting the tubes in the centrifuge, wasting the flow through from the columns and so on. In addition, the QIAcube HT has a chain of custody. So he knows exactly, and you know exactly on the audit trail, if everything went right. So we use the samples with the UCP multiplex master mix and some standards to amplify the 16S gene. Looking first on the left side, which is our kind of quality control, the non-template controls were clean, so we had no background, and we could get down to 10 femtogram of bacterial DNA. Yeah, the bands here are a bit weaker. However, because of the high bacterial load of stool samples, we have just done 34 PCR cycles. So see it from another perspective. Even with 34 PCR cycles, we get down to 2 E. coli genomes. On the other side, all 16 stool samples which were isolated worked perfectly without a sample normalization prior to the PCR. So we just used 1 microliter of the of the eluate from a QIAcube HT, which is a perfect workflow integration. So I said before that the Earth microbiome, for example, is very powerful and is used a lot for 16S sequencing. However, also not all bacteria can be identified with this primer pairs, or labs have other needs and want to have a perfect solution out of a box. For this, we have developed the QIAsec 16S 18S library kit. This kit gives you the power to choose the 16S regions you want and to combine them with a fungal detection. So the kit has two different formats. You can either have a screening panel, which consists of 3 duplex PCRs for 16S covering the entire 16S region from V1 to V9, and also a multiplex for ITS in this. When you have done this, or when you, for example, know already which regions are interesting for you, you can order specific regions. So this means you would have only one duplex or one triplex PCR with 2 16S regions plus ITS. In addition, the kit includes everything you need. So you have your primers, master mix, water, and beads for the entire workflow up to NGS. You can get this with 24 or 96 different indexes, so you can do up to 96 samples in one run from the beginning to the end directly on the MiSeq. So that's the structure. What are the benefits beside having the freedom to choose the primers you want? We have a phased primer system. So this means that in the primers are additional bases, and by this you are shifting your position of the 16S genes between 1 and I guess 9 or 10 nucleotides. This has the benefit that it's massively increasing your heterogeneity in your libraries and improves your cluster calling on the MiSeq. The beauty about this, you can eliminate phi-X. So this decreases the sequencing cost a lot. And the base quality is really improved by this. So, as said before, the number of PCR cycles is important for your bias and also for amplification of background. Here we have had a potential or a special interest on looking on a system which can amplify with low PCR cycles from even very low DNA inputs. So for 1 nanogram of DNA, we're taking 12 cycles of 16S. When you go down to 1 picogram, you have 20 cycles. It works perfectly. In addition, we have a smart control in it, which you can amplify with the 16S primers. On the other side, it's an artificial system, so you can use it as your negative control for the sequencing result and as a positive control for your library construction. Here is a short overview how the kit works in principle. You have to extract your samples, do your 16S PCR. When you have the screening panel, as mentioned before, you have 3 reactions per sample. You have to do 2 bead cleanups, pool them, do your indexing PCR with the sample indices, do the bead cleanup, and then you have your library QC. This is a really streamlined and quick process, which allows you to standardize your sample process a lot. So now you've done everything. Your libraries are sequenced, you get the sequencing results back, and now you have to analyze them. As said, my challenge when I got my first NGS results was just the pure mass of results and that I'm not a bioinformatician. So at the time when I got it, unfortunately, I wasn't aware about CLC because I was still at the university, and I would have loved it. Because CLC is an integrated analysis which delivers all the data you need. So it can do the complete analysis of microbial genomics and metagenomics, and it's completely integrated. And you can use it for what you want. You have streamlined workflows to focus on the interpretation of the results. And the algorithms allow you to run the analysis on a standard computer. So you don't need the multi-core, high-power server, which was needed before. So how does it work? You have your CLC Genomics server and your CLC Genomics workbench. You load your data in, and then you pick the plugin you want. So for example, when you look for 16S, you can use your CLC microbial genomics work module, and in the end, get the results in the way you need them. You can look, for example, for microbiome analysis, the contribution, ag bio and food safety, or also look for outbreak analysis. I have here a bit more detailed overview. As said, we start with the data import, then you do the trim, or the software does the trimming and the quality filter of the raw reads. It will merge the paired end reads, so the forward and the reverse read for your sequence, and then you can use the database you want. For 16S, you can either choose between the green genes or the silver database. When you have whole genome assemblies, you take the gene ontology of FAM. Based from this, here you would have the de novo assembly for the metagenome analysis for whole genome sequencing. On the other side, for 16S, you would have the OTU clustering and the tree building. And as the secondary analysis, you look for the alpha and beta diversity to get your phylogenetic diversity in your sample and to get some statistics out of it. Not to forget, the software is giving you directly publication quality visualization plots for your data. As said in the beginning, I would like to end my talk with showing you some application data. During the last decades, I have to say, more and more people were looking for aging and the impact of aging on the human being. In the beginning, people like Hayflick, for example, looked on the human cells and looked what happens to the cells. So, for example, Hayflick identified the Hayflick limit, which is, in the end, the cellular senescence. It was found that cells, when they're aging, getting more and more genome alteration. So the genome instability is increasing. Telomere attrition is seen. So your telomeres are shortening and your cells cannot replicate anymore. Your methylation patterns are changing. So you have epigenetic alterations in your DNA. And there are many other processes in your body which are affected. However, in the last time, as for pretty every area of interest, the microbiome came into focus. So it's an increasing focus on the gut microbiota as a potential regulator of aging. So we wanted to have a look on this. We used stool samples from 10 persons. 5 of them are in a higher age, 5 of them are in a lower age. As you can see, the distribution was roughly 50-50 between male and female. And yeah, we used 200 milligram of human stool, lysed them with power bead tubes for 10 minutes on a vortexer. And the supernatant was then placed in a Kaya cube, and using the Kaya Empower fecal DNA kit, the DNA was isolated. Here you can see a picture of the DNA samples after isolation, and we succeeded in amplifying good amounts of high quality DNA. When we look at the DNA yields for the samples, it seems like the young people in general had a higher DNA yield out of the samples than the old people. So the next step, and I will first show you a small overview. The next step after the DNA isolation was the amplification of the 16S sequence to prepare everything for NGS. Here I would like to show you the overview of the entire 16S sequence, and the plot is giving you the different high constant or high conserved regions. Based on this, we have decided to use the V4, V5 region, which is in the end one of the most common used amplicons for 16S sequencing. 50 nanogram of inhibitor-free DNA were used, and 22 PCR cycles were done with the modified Earth microbiome primers. Here are the PCR products, so we succeeded to amplify a sufficient amount of 16S amplicon for each of the samples. And not to forget, here the blanks are empty sample preps on the QIAcube HT, so we have no background coming from the kit. PCR products were then evaluated from the gel and used for the QIAsec one-step amplicon kit. Subsequently, these libraries were normalized and used for Illumina sequencing with a paired end 250 base pair sequencing. So up to this point, we have no issues with the workflow. The inhibitor removal was efficient, could perfectly amplify our 16S regions, and we had no background. After the NGS run, we got some sequencing statistical results. As you can see here, we have generated roughly 22 million paired end reads. We had 90% of the reads passing a quality filter of Phred score of 30. 88% of the reads could be assigned to one of the used indices, so most of the NGS reads could be used. And we had a pretty good distribution, so 9 out of the 10 samples had around 10% abundancy in the reads, so we had a pretty good distribution of the samples. So after running the data in the Microbial Genomics Pro Suite, this is the first plot we obtained. This is giving you the results at the genus level. And as already mentioned here, you can customize your outputs in the way you want them. And also reads which were not mapped directly were then clustered and de novo analyzed. So here already, on the left side the old and on the right side the young individual samples, it seems for me that we have a higher variation or a higher diversity in our young individuals, as you can see here. Many more different bars compared to the older individuals. So as a secondary analysis, we looked for alpha and beta diversity. Here the alpha diversity plot clearly confirms this feeling. All young individual samples had a higher diversity than the old individual samples, confirming that with age you have an effect on your bacterial diversity. As a next point, we wanted to know if this is significantly different. So we did a Permanova analysis, or the Microbial Genomics Pro Suite did this analysis. And as you can see here, we have a significant difference between the young individuals and the old individuals. So by this data, we could show that aging has an effect on your microbial community in the gut. We can, of course, not say if the reduced microbial community variation is a source or is caused by aging, or if this is affecting aging. But studies on different dietary preferences, also, for example, in Japan or other blue regions in the world where people get very old, indicate that the dietary conditions have a strong effect on aging. These were the data I wanted to show you, and in summary, here are the kits I was talking about in case you're interested and would have a look on this workflow on your own. Thank you all for your attendance, and I hope I could explain this workflow and to you in an appropriate manner and highlighted some points which might be interesting for you. Thank you.
QIAGEN.
Sample to insight.